Okay, so it looks like our next question falls on the next page. So letter D up here at the top, still that same line of questioning with the test cross. So letter D, offspring of the cross above, which would be that test cross we just completed, yields 24 offspring. So 24 little mice babies. 13 of those babies have black fur, 11 have white fur. Okay, so 13 with black fur, 11 with white fur. What was the genotype of that wild black mouse? What was the genotype of the parent? Big B, little b, right? It has to be carrying the white allele, the recessive, because that's the only way some of the babies could have that lowercase b, have that trait. It had to come from both parents if there's some white fur. All right, let's jump to dihybrid crosses. So number 4, this is one of the big ones. This is as big as we'll go in this class. So in a dihybrid cross, first off, let's break this word down. Dihybrid, di. Di means two. So we are working with two separate independent traits. Two separate traits. Monohybrid, one trait. Dihybrid, two traits. Here we go. In humans, there's a disease called phenylketonuria, which is caused by a recessive allele, abbreviated PKU. It's caused by a recessive allele. People with this allele have a defective enzyme, cannot break down the amino acid phenylalanine. This disease can result in mental retardation or even death. Let capital P, all right, here we go. It gives us a key. Let capital P represent the normal PKU gene. Okay, so I'm going to come over here to the side and I'm going to make myself a key. It just helps me keep my traits organized and straight in my brain. What letter represents what trait? So I'm coming over here and I'm going to put a capital P. This is normal. Now I'm using a P just because the name of that disease starts with a P. So I'm not using an N for normal. I'm using the P to represent that trait. Capital P means they're normal. They do not have that defective enzyme. Okay, which means a lowercase p would be PKU. That's the trait. They're carrying that trait for PKU. There. I don't know if you guys can see this on your, I'm kind of getting off, my brain's getting off track here on this video. I'm seeing a thing in my corner. Looks like a thumbnail or something in the corner of my screen. I don't know if you guys can see it on your screen or not. But it's distracting. It's distracting to me. It's covering up some of my questions. So I'm having to move things around a little bit. So apologies if I'm giving you motion sickness again, because that little thumbnail that's there is blocking the question and I can't move it out of the way. So I'm kind of getting distracted. Okay, back to it. Capital P, normal, lowercase p means PKU. All right, also in humans, here's our second trait, dihybrid cross. Also in humans, in a condition, also in humans is, I think that's a typo, also in humans is a condition called galactose intolerance or galactosemia, which is also caused by a recessive allele. This tells us let G represent the normal allele for galactose digestion, so a lowercase g would be galactosemia. All right, so here we go, making our key here. G is normal. And then lowercase g, because it says it's recessive, a condition called galactose intolerance or galactosemia, which is also caused by a recessive allele. Yeah. So lowercase g is galactosemia. Galactosemia. Galactose is a carbohydrate. These people have trouble breaking. It's like lactose intolerant. These people are galactose intolerant. Just a different type of carb, different type of sugar that's there. All right, we've got our key. We're set up. Question A, a parent, heterozygous for PKU and galactosemia, is crossed with a parent, heterozygous for PKU again, but has galactosemia. What are the genotypes of the parents? All right, a parent heterozygous for PKU. You know hetero means different, so we're going to have a capital P and a lowercase p. And galactosemia, so heterozygous for both. So this one's going to be big G, little g. There's one parent, heterozygous for PKU and galactosemia. Is crossed with a parent heterozygous for PKU again, which would be capital P, lowercase p, and who has galactosemia. If they have galactosemia, that's lowercase g. So if they have it, it's got to be 2 little g's. There we go. Okay? All right, we got our parents. Show the Punnett square for this cross below. Now this is where students start to mess up. So I'm sorry I'm giving you motion sickness again. I'm gonna move this up just so I have a little more space to work with on my side of the screen. Okay. Sorry. I can't get it straight, but maybe that's close enough. Okay. I want to show you what not to do first because this is what I see when students make mistakes. 90% this is the mistake they make. Okay, don't do this. I'm going to erase it, so don't follow this, but they do this: P P G G. That's one parent, and then they'll kind of do the same thing across the top. This is wrong. Big X. Do not do this. Okay, why shouldn't we do this? Because this is a dihybrid cross. What does di mean again? 2. How many traits are you showing right here for one parent? 1. You're just showing the normal allele for PKU. 1. This is wrong from the get-go, okay? Because it's a dihybrid cross, you're dealing with 2 traits. That means you have to have each breakdown of the parent, each breakdown of these alleles has to have 2 traits in it. So this is incorrect from the start, okay? So here's what we're going to do. We're going to pair up these alleles. We're going to go capital P, capital G, okay? Capital P, capital G. Then we're going to pair up capital P, lowercase g. All right, and if there were any more alleles in the chain, if we were doing, you know, trihybrid, quadhybrid, we would just keep going and we would pair up all these alleles. There's nothing left for my big P to pair with, so now we're going to jump to little p and G. All right, do you remember earlier I mentioned I like to put capital first because it's dominant, keep my alleles trait? This trumps that rule because first and foremost, it supersedes that rule. First and foremost, you need to keep your traits in order because you're sure going to go crazy if you start getting your traits out of order and then you're trying to interpret genotypes and phenotypes. So what is above putting capital first? Keeping your traits in order. That supersedes. So I'm going to always have my P's first, even if that's a lowercase p. Okay? All right. Then I'm going to pair up my P with my G. Lowercase. P, lowercase g. Okay, there's one parent. Let's do our other parent across the top. Capital P, lowercase g. Capital P, lowercase g, the second one. Then I'm moving to the second P, lowercase g. And then I'm doing the last one, lowercase p, lowercase g. All right, I got my parents in. Now we're ready to fill in our dihybrid cross. P's first, G's second. The rule that supersedes capitals, you know, first is always keep your traits in order. So trait, what's coming first? The PKU trait, that's coming first. So I'm going to go capital P, capital P, capital G, lowercase g. Capital P, capital P, capital G, lowercase g. Capital P, lowercase p, capital G, lowercase g. Capital P, lowercase p, capital G, lowercase g. There we go. Next line. Capital, capital, little, little. Capital, capital, little, little. Capital, little, little, little. Capital, little, little, little. Here we go. Capital P, I'm putting capital first. Lowercase p. Capital G, lowercase g. Capital P, lowercase p. Capital G, lowercase g. Two little p's. A big G and a little g. Two lowercase p's, a big G and a little g. Last line. Capital P, bringing it down. Lowercase p, bringing it over. Two little g's. Capital P, lowercase p, two lowercase g's. Two little p's, two little g's, two little p's, two little g's. Done. We got it filled in. Okay, we'll scroll up. Here comes the motion sickness. Apologies. Let's look at these questions. What are the chances of having a child that's completely normal, does not have either disease? Now remember, I scrolled up and I'm out of the view of my key over there, but remember, capital P was normal, capital G was normal. So our question says, what are the odds of being completely normal? So we're looking for anything that has a capital P and a capital G. So let's count them. I'm going to use a highlighter, I think, tool so that we can see this. All right, here's a capital P. It's not very thick. Let me increase my thickness. All right, here's a capital P, capital G. That's normal. Here's normal, here's normal, here's normal. No, that's not normal. Undo. I jumped ahead. Why is that not normal? Because I've got two lowercase g's. They're going to have galactosemia. So I'm not going to highlight those because it says completely normal. Does not have either disease. Right? Does not have, whoops, I'm in the highlighter realm again. Does not have either disease. Not have it. I'm getting my tools mixed up here. Pen. I needed the pen. There I am. Does not have either disease. Okay, so I'm not going to select the anything with lowercase g's. Those are not normal. Here's another set. Here and here, here and here. Nope, not doing this lowercase p's because they've got PKU. Not doing that. Not doing that. That's it. That's all I got. Okay, so let's count them. One. Let me do a tally. Undo. 1, 2, 3, 4, 5, 6. So I've got 6 out of 16. 6 out of 16 having a child that's completely normal. Just leave it as fractions. When you get into the dihybrid, trihybrid, these big squares, just about need a calculator to work out some of these percentages. You can do it, work it out if you want to. 6 out of 16. Okay. What has, part B, has just PKU? What percentage has just PKU? So that means the G's would be normal and the PKUs would have the lower P's. All right, so I'm going to highlight these maybe in a different color. Has just PKU. Nothing with a capital G fits this. No, that's wrong. Has just, we need capital G's. You see how confusing this gets? You have to think about this, really. Has just PKU. So we're looking for lowercase p's and capital G's because capital G would be normal. Okay, lowercase p's. Here we are. Right here, lowercase p's. And notice there's a capital G. So this is, they do not have galactosemia. They only have PKU. This one, that's it, 2. 2 out of the 16. I'm going to maybe highlight this one in green. Has just PKU. All right, maybe that'll kind of give you a visual cue for that. So 2 out of 16. Which ones has just galactosemia? All right, so here we go. Just galactosemia. That means they're going to be normal and have the big P's, but they're going to have galactosemia and have little g's. So we are looking for little g's with a capital P. Here, 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 here. All right, I think that's it. Has just galactosemia. So we're looking for only the little g's with capital P's. So there we are. Got those highlighted. I'm gonna highlight this in blue so we know what you're looking at there in the Punnett square if you happen to go back and rewatch this video or grab a screenshot or something and practice. All right, how many we got? 1, 2, 3, 4, 5. Nope, 1, 2, 3, 4, 5, 6. 6 out of 16 here. Okay, and then what's gonna have both diseases? Well, that's gonna be your two littles at the end there. So it's gonna be this one and this one have both diseases. So 2 out of the 16 here. My handwriting's kind of bad. 2 out of 16 for there. Now, here's a quick way to check to make sure we didn't miss something. I kind of highlighted as I went so you can tell visually that I didn't miss anything, but you doing it out on paper, maybe with just a pen or pencil or stylus if you're on your iPad or something, count them. So 6, I'm counting these right here. 6 plus 2 is 8. Plus 2 is 10. Plus 6 is 16. That would be 16 out of 16. We didn't miss anything that way. When you add up that, if you're like, wait a minute, I've only got 14 out of 16, you're like, I skipped 2 somewhere, and you go back and count and check your answers there. But there's a dihybrid cross right there, my friends. Takes a little bit of thinking on it, a little bit more complex because you're dealing with 2 separate traits. Okay, I'm fixing to slide up again because now we're jumping to incomplete dominant, so sorry for the motion sickness. All right, incomplete dominance. Here we go. What does incomplete dominance mean, by the way? If something is incomplete, well, that means you do not have a dominant trait. These 2 traits will essentially blend. Black, white make gray. Red, white makes pink. Red, orange makes, red, yellow makes orange. Red, yellow makes orange. So in incomplete dominance, you do not have a trait that masks the other. Instead, they're both expressed in a blending fashion. They blend, okay? Okay, let's look at number 5. 4 o'clock flowers exhibit incomplete dominance. 2 pink, oh, here we go. 2 pink 4 o'clock flowers are crossed. Complete a Punnett square for this cross. So let's make a key first, okay? Red, we can do this one of 2 ways. The way I teach it and the way I present it, I still use the same letter for this. But I have seen it done with 2 separate letters. I just don't. So I'm going to show you a little bit of both ways. I'm just going to say red is R. Lowercase red is probably white. And then when you mix them, an R and an R equals pink. Okay? I have seen individuals do this with the different letters. R, lowercase, because it's incomplete dominant, you really don't have one dominant over the other. Lowercase r equals red. Lowercase w equals white. And then rw equals pink, because that's the heterozygous form. We're mixing. Either one of these is fine, as long as you know what your letters are representing. Your questions are always asking you about genotype and phenotype of offspring. as long as you communicate your key, you know, as long as the heterozygous is the blending of that, of your trait, it's really not a huge deal as to how you create your key. I'm just going to do the R and the W and the RW for that, just because having them all lowercase is maybe a clearer picture of being incomplete dominance. But again, when you start getting into dihybrids and trihybrids, if you use the same letter per trait, that's just easier. In this class, we don't go that much in depth. You go on to a senior college and you're taking legit genetics class, then just follow the instructions of your instructor at that point with it. Let's take a look at this. 2 pinks, 4 o'clock flowers. So they're going to be RWs. So I've got, my parents would be RW times RW, or if you did the big R and little r, totally fine. So we're going to have red and a white, and we're going to have a red, we're going to have a pink, we're going to have a pink, and then we're going to have a white. Okay, and I just kept my sequence consistent. So notice I put R first in all my offspring just to stay organized, easier to interpret that way. All right, there's our Punnett square. What is the predicted genotypic ratio for the offspring? So genotypes are your alleles. So it's asking for a ratio, not a fraction and not a percent. So I'm going to have 1 lowercase r to 2 RWs to 1 WW. There's my genotypic ratio. What's the predicted phenotypic ratio? Again, a ratio. So I'm going to have phenotypes, what they look like. 1 red to 2 pinks to 1 white. All right, genotypic, phenotypic ratios.